So welcome Sam Hedges for our NPA seminar today. He's going to be talking about imaging nuclear recoils with light sheet microscopy. Sam does PhD at Duke with me in the Phil Barbeau group, working largely on coherent, where he won the Springer Thesis Prize for measuring a number of neutrino interactions on lead and iodine. That won't be the subject of today's talk, but then he spent some time at Livermore working on NEXO, and now he finds his way over to Virginia Tech working on a combination of this work as well as coherent as the analysis coordinator and a few other projects. So yeah. I'll leave it to you.
All right, thanks, Tyler. So as I said, if you don't know me, my name's Sam. I'm a postdoc at Virginia Tech, and I've been there since February. And what I've been working on is what I'm going to present today, and this is on imaging nuclear recoils with light sheet microscopy. So the way this talk is structured, I'm going to start off by giving an overview for why we care about detecting nuclear recoils, and then what are the advantages of looking for nuclear recoils through crystal damage. I'll then talk about some early efforts that have been done towards detecting damage tracks. And then I'm going to focus on the technique that we're using, which is using color centers to image the damage done to crystal lattices through fluorescence microscopy. And then finally, I'll present some of the ongoing work in our lab. So starting off with nuclear recoils. As many of you know, nuclear recoils are one of the signatures of the elastic scattering of particles like WIMPs, neutrinos, or neutrons. They all produce the same detectable signature. And the typical way that you try to measure these signals are through things like light in scintillators, charge in ionization detectors, or heat in bolometers. But in all of these materials that I've just shown, in addition to those channels that you're typically going after, you can also have crystal damage occurring as a result of the nuclear recoils. And generally, it's difficult for gammas to create the same kind of defects as the nuclear recoils from neutrinos, neutrons, and dark matter. So these are more sensitive to those heavy particles than photons. So if we want to go ahead and look at this damage, we have to give the nucleus some energy. And in addition to doing that, we actually have to give it enough energy that it doesn't immediately recombine in the site that it was just knocked out of. This threshold is material dependent, and it's typically on the order of tens of eV or so. And when we have a nuclear recoil that produces a vacancy, that ejected nucleus typically will then interact with other nuclei, and what we'll get are some damage tracks like you can see here. This is done with SRIM. This is a plot of the vacancies of a couple lithium-7 recoils in lithium fluoride. So you see these nice tracks from nuclear recoils. This all works because these tracks can persist at room temperatures over long periods of time. And if we can measure this damage, then all of a sudden we have this new channel for going after nuclear recoils from rare processes like dark matter neutrinos. So I think many people here are aware of dark matter. Just to give a brief overview, there's a variety of different astrophysical observations that suggest that there is additional matter present that we can't see. These come from things like galactic rotation curves, where you look at the distribution of speeds of stars as you go further out in the galaxy. And based on the visible mass, you expect this one distribution, but in reality, you see a different one. Another good example is the bullet cluster, where you have most of the mass existing in these, the visible mass existing in these pink areas, but the blue areas show the true mass distribution from gravitational lensing. And both these measurements, along with a variety of others, are all consistent with the idea that there's some additional matter present that's about 5 times more prevalent than visible matter in our galaxy, or in the universe. So one potential explanation for this is that this dark matter is a particle, and it could be a variety of different particles. One of these candidates is the WIMP. And while we have all of this astrophysical evidence for this additional mass, we've never observed this particle here on Earth. The typical approach if you're building a WIMP detector is you get a large detector, you make it very radio pure, then you put it deep underground and then wait for something like 10 years. So the exposures for these detectors are typically on the order of 10 ton years. Shown here, this is an exclusion plot from a recent LZ paper showing where the limits are for xenon right now. So you have this one approach of taking a ton of material and waiting 10 years. You can actually get a very similar exposure if you take 10 grams of material and you wait 1,000,000 years. And this is the basic concept behind paleodetectors, is that there are minerals and rocks that have been underground, well shielded for millions, if not billions of years. And in that time, they've potentially integrated a lot of really rare events that could have occurred in them. If you're trying to use this technology, you have a different set of challenges. Some of these are shared with the more active approaches. You have to have low environmental radioactivity. You still want these to be at sufficient depth to shield from cosmic rays. Additionally, though, you have to understand how these samples move over the course of their life. On geological timescales, things don't necessarily stay in place underground. They can actually come up to the surface and go back down. You also have to understand the temperature that the rock has been exposed to, as the damage can be annealed if it reaches a sufficiently high temperature. And all of these challenges, none of this has anything to do with readout, which is another huge challenge for this technology. Shown here, this is some exclusion plots for different minerals with some assumptions about the exposure and the sensitivity of the readout scheme. You can see that these are pretty similar to the xenon limits, and these are looking at something like 100 kilogram mega-years exposure. Aside from dark matter, another application of nuclear recoils is reactor neutrino detection. So there are something like 10 to the 20th neutrinos emitted per second from a 1 gigawatt reactor. And if we can study these neutrinos, we can learn about the reactor power, the status of it, the different isotopes in the core that are fissioning. The typical way that we go after detecting reactor neutrinos is through inverse beta decay on hydrogen, which is shown here. We have an electron antineutrino interact with a proton, creating a positron and a neutron. The positron will deposit some energy and then annihilate. And then typically tens of microseconds later, we'll have this neutron capture. And this coincidence gives us a powerful background rejection, allowing these detectors to operate at the surface. So while all of this is really nice, IBD is only sensitive to neutrinos with energy greater than 1.8 MeV. And that only encapsulates about 30% of the total flux of neutrinos emitted from nuclear reactors. And in particular, there are some components from neutron capture on non-fissile components around the reactor that produce exclusively low energy neutrinos. And one potential application of this is detecting breeding blankets. So a breeding blanket is where you'll have some amount of uranium-238 surrounding the reactor core that can be used to breed weapons-grade plutonium. And the signature of that is this dotted line shown here that ends at about 1.26 MeV. So if you want to see if somebody is doing this, neutrinos can be a signature of it, assuming that you have a low enough threshold for this process. One of the ways that you can go after these lower energy neutrinos is through a process known as coherent elastic neutrino nucleus scattering, or SEVNs. This is a standard model neutral current process in which the neutrino interacts with the nucleus as a whole. For this to happen, the momentum transferred to the nucleus has to be comparable to the size of the nucleus, which means that this occurs for low energy neutrinos, typically less than 100 MeV or so. It has a large cross section, which is expected to scale with the number of neutrons in the recoiling nucleus squared. But the only detectable signature is this low energy nuclear recoil. It was predicted in 1974 for the first time, but evaded detection for more than 40 years before it was first observed in 2017 using pion decay at rest neutrinos. And then very recently this year, we had the first ever observation of reactor SEVNs in January. So we can use persistent track detectors to try to study SEVNs interactions. That's because, as I mentioned, they have these low thresholds on the order of tens of EV. And one of the other advantages of these is that they're passive detectors. You go deploy your massive target, wait, bring it back. You don't have to have active electronics or liquid nitrogen deliveries or anything like that, which can be difficult to arrange at a reactor. All right, so I've given you some motivation for why we're trying to detect nuclear recoils. You might ask, have people done this before? And the answer is yes. So this is an old concept. Some of these first ideas for looking at tracks in solids from particle interactions happened in the 1950s. The general approach of all of these early efforts was to create some large energy depositions, such as fission tracks, or they take these samples to accelerators where they'd expose them to hundreds of MeV ions, or they would look for high Z cosmic ray interactions. That would damage the material. Then they would take that and slice it into thin slices and etch away the tracks. The damaged regions would actually etch faster than the non-damaged regions, and that allowed them to then blow up these tracks to a large enough size where they could image them with an optical microscope or a TEM. There are some drawbacks of doing this. One, it's destructive, which means you don't really want to do this for rare samples. And also, the big thing is that it has a limited throughput. You can only scan very small areas of these materials, which means that you can't do some of these rare event searches because you just can't scan enough mass to do that. And then also these early approaches were insensitive to lower energy depositions, so they couldn't see neutron-induced nuclear recoils or alpha depositions. Nevertheless, this was really cool at the time. There were lots of studies that used this technology to do things like fission track dating, where they would tell the age of minerals by the number of fissions that occurred. They would study heavy cosmic rays in meteorites and fossils, and I think they even did some lunar samples with this technology. There also were searches for things like magnetic monopoles. Things really changed in the 1990s when you had the first searches for WIMP dark matter using damaged tracks. So this is from a paper from Snowden-IFT, where they used an atomic force microscope to scan mica in a grid. And with this, they were able to achieve sensitivity to these lower energy signals. So they were able to see alphas and neutron-induced recoils for the first time. This process was pretty similar. They had to cleave and then etch their mica samples as they did before. But in this case, they then took an AFM tip and dragged it across their sample in a grid with 150 nanometer pitch. From that, they were able to measure the pit depth and use that as an estimate of the energy. And they were able to set some of the first limits for WIMP dark matter using this damage track approach. Shown here, this is one of the images after processing that they've seen. And they have these various damage defects along with the depth. And yeah, they were able to set limits that are probably not competitive with PALET today, but at the time these were an interesting result. So since then, there have been many new ideas for how you can detect damage tracks. You want sensitivity to these lower energy signals as well as a high throughput. So there are things like X-ray microscopy, strain imaging. There's multi-scale imaging where you'll use some kind of coarser approach to find the interesting regions of your target, and then use something sensitive like AFM, TEM, or SEM to do a fine scan of that region. What I'm focusing on today is fluorescence microscopy. In particular, there are a couple of different ways that you can image samples with this. I'm going to be focusing on selective plane illumination microscopy. And in general, yes, there have been some technological improvements that make these different imaging techniques possible, but we're also benefit greatly from this increase in storage space and computational power. If you're trying to scan a cubic centimeter with micrometer or nanometer scale precision, you generate a lot of data. And just to help show some of this interest, for the past couple of years, there's been an annual workshop on the mineral detection of neutrinos and dark matter, and you can find the most recent proceedings from that here. So the technique that we're focusing on is using fluorescence microscopy to image color centers. So I should first introduce what color centers are. And what this is when we have one of these vacancies from an ejected nuclear recoil, in some materials, electrons can get trapped in these vacancies and turn those sites optically active. Essentially, this is just a basic quantum mechanics problem. You have an electron trapped in a potential well, so it can be excited to discrete states and then de-excites by emitting photons in a process known as fluorescence. This is actually surprisingly common. It occurs in a number of naturally occurring minerals, such as diamond, olivine, quartz, and the other one shown here. And it also happens in a lot of very common scintillators. This isn't something that you necessarily need those advanced instruments to see. You can actually visibly see this if there's a sufficient density of these color centers. So shown here, these are diamonds with various defects. You can see how this affects their color. So in particular, these ones down here that have vacancies from some kind of radiation damage done to the diamond, that can actually change this from this white color to this greenish yellow color. by forming these NV vacancies. So there are things called green diamonds that are naturally occurring. They're pretty rare because they have to be grown in an area where there's sufficiently high radioactivity. But these occur, and they're actually very expensive if you want to get one of these naturally occurring ones. For most of the work that I'm going to be presenting today, I'm not looking at any of those previous materials I mentioned. Instead, I'm going to be talking about lithium fluoride. This has some advantages. So lithium fluoride color centers have been well studied. At one point in time, there was a whole branch of effort to try to develop optics with these color centers. The color centers can be excited with visible light. The defects are stable at room temperature. The material is only slightly hygroscopic, which isn't true for some of the other materials, which means we don't need casings. We just need to make sure these aren't exposed to too much moisture. And then they anneal around 350 Celsius. So you can reset these crystals back to this pre-damaged state by heating them in something a little bit hotter than your home oven. Additionally, one of the things that's really nice about lithium fluoride is that lithium is around 7% lithium-6 naturally. And with that, you have this in situ calibration capability from thermal neutron capture on lithium-6. I think as many people know, when you have lithium-6, it has this really high thermal neutron capture cross-section. When this happens, you get a back-to-back tritium atom and an alpha particle, and those are monoenergetic. So we can use this to help calibrate what we see in our detectors. Prior to doing any kind of imaging to look at individual color centers, one of the first things that we did is look at bulk fluorescence properties. So essentially, what we're doing here is exposing our crystals to a lot of radiation to generate a high density amount of color centers. And then we're just measuring the total fluorescence light that's emitted from it when we have some excitation. So for this, we used a Curie adjuvant spectrophotometer, which basically we just have a xenon lamp. We can select both excitation and emission frequencies, and we can scan over the intensity of the fluorescence as a function of wavelength. So we took some cubic centimeter sized lithium fluoride samples. We exposed these to neutrons from an ambineutron source, as well as gammas from a cobalt-60 source. And with that, we were able to see some bulk fluorescence properties. So shown here, this is the emission spectra after exposing these crystals to 445 nanometer excitation light. And you can see that there are these two different fluorescent bands. So this F3, this just means that these are 3 vacancies in close proximity. The F2 means that there are 2. That produces a different potential well, so you get different excitations. And one of the things that's interesting here is that the ratio of those 2 bands changes depending on the type of irradiation. Additionally, we did a measurement where we actually put this sample into the sources and recorded the fluorescence while we were irradiating it with some optic fibers. And with this, you can see that the fluorescence grows much more significantly for the neutron irradiation than for the gamma irradiation. Turns out that these are about 50 times less sensitive to gammas than neutrons. So we typically use this kind of bulk fluorescence techniques when we're trying to study what are the excitation bands that we need to use, what are the emission bands, and to do things like look at how these the fluorescence from these samples changes as you heat them, as you begin to anneal the damaged tracks. But now going on to actually trying to image these individual color centers. So there are a couple of techniques that have been used in the past. Typically, people use either wide field or confocal microscopy to image color centers. With wide field microscopy, we're essentially just uniformly illuminating the entire sample and then taking a picture of it. So with this, the imaging speed is very fast, but the resolution isn't good. And that's because you have light produced from behind the sample that then gives you blurry images like the one shown here. This also means you have to have thin samples. They're typically less than a millimeter or so, and you can only image a few tens of micrometers into the sample. The other approach that people have used is confocal microscopy. This, instead of doing this imaging the entire sample at once, you're actually imaging point by point as you scan over the sample. As a result, this is much slower, but you have much better positional resolution. You can also go to deeper depths and deal with slightly thicker samples. So people have done this to look for color centers in lithium fluoride. On the left here, this is an image taken with wide field fluorescence microscopy. On the right, this is using confocal microscopy. And you can see these tracks here. It's a little bit blurry on the TV screen, but this is after exposing it to thermal neutrons. So you get some of these back-to-back captures like you can see here. On the right, this is from exposing the lithium fluoride to protons, 2.6 MeV protons, and you can see more point-like defects. But with both of these are just looking, barely into the surface of these samples, and the samples are very small, which means that you really can't use these approaches to scan a large volume of material. So instead, what we're focusing on is called SPIM, or selective plane illumination microscopy. With this, we essentially have a light sheet, and our sample passes through that sheet. As the sample moves, we take pictures of the fluorescence in each slice, and this allows us to image 3D volumes of material. The samples are in a fluid that matches the index of refraction, and that's so the optical path doesn't change as we move the sample away from the camera. And as a result, we can do much faster imaging. We get a similar positional resolution to the wide field approach, but we can also look through thicker samples. So we're looking at samples that are on the order of a cubic millimeter or so. And this instrument is used by biologists. They look at larger samples with this. So the instrument that we use is called the benchtop mesoSPIM. So this is an open source light sheet microscope, meaning you can find the instructions and parts list to build one of these yourself online. With this, we get around 1 micrometer lateral resolution and around 5 micrometer axial resolution. That comes from just the thickness of the light sheet. And with this, it takes around 1 to 2 hours to scan a cubic centimeter of material, which is much faster than any of the previous approaches. It also generates about a terabyte of raw data per cubic centimeter. So you can see why this maybe wasn't possible 20 years ago. Shown here, this is an image of a mouse brain in the microscope, and then here is one of the slices of this image. So this is used by biologists a lot. I think we're the first group to have one dedicated towards particle physics measurements.
How big are brains?
So I think this cuvette is 5 centimeters thick. So that's probably a centimeter and a half, 2 centimeters, something like that. But yeah, because this is open source, right? People can modify it. People are looking at larger samples with this. There's a group that's trying to image an entire human brain in slices using this approach. So yeah, this is used a lot by biologists. And one of the advantages of this being open source is that with some of the existing light sheet microscopes, you're locked into the software that is used, and you can't really make modifications. It's hard to then get the data into a format where you can run your own analysis on it. So we've used 2 of these benchtop mesoSPIMs. The first one was at the University of Zurich, and this is one of these instruments that's primarily used by biologists. So we were able to get a couple weeks of time on it, but we weren't allowed to really make any modifications to it. We couldn't change the filters or any of the hardware or the camera. So as a result of that, we decided to build our own microscope over the summer at Virginia Tech. So that's what I've been working on. And before we talk about the actual data we've collected with that, I wanted to just give you a brief overview of how this works. So this maybe looks a little bit complicated, but it's actually pretty simple. So we start off with an objective lens that focuses our laser into our sample, which is located over here. And this is just an off-the-shelf camera lens that can focus this down to a beam waist of about 5 micrometers. This is what sets this axial resolution that I talked about. We then have this electrically tunable lens, and what that can do is that can drag the beam waist left and right across the field of view of our camera. The way that this kind of trick behind how all this works is that the camera also has a rolling shutter. So you're only exposing the column of pixels that you currently have the waist centered over. And that way you're getting the highest signal to noise ratio and also the best axial resolution. So you can imagine, right, this moving across the screen and then the line of pixels being exposed and read out is synced with that. So when you do that, instead of getting this thing that comes down to a waist, you get just a nice straight line going across the screen. Then we have a scanning galvanometer mirror. So that oscillates the laser up and down much faster than the camera exposes things. So the camera essentially just sees a uniform sheet of light, and that's where the light sheet actually comes from. And then we have, of course, our magnification lens and our camera. So here are some pictures of some of these samples in the mesoSPIM. So you can see it's this little cube right here, transparent material on this 3D printed holder. And on the very right here, this is, while we're exposing it, and you can see it's kind of glowing this yellow from the fluorescence from the laser light that we're using. So, wow, this looks even worse on the TV than on my screen. So this is one of the first images that we took with the mesoSPIM at the University of Zurich. And if you kind of squint here, you can see there are some spots that are brighter than others. So with this, there's lots of noise in this, but we're able to do some image analysis and come up with something that looks like this. So that involves basically some filtering, some normalizing, and some thresholding. And we get things that have some nice tracks in it. So we have samples that have been exposed to fast neutrons from our ambi source as well as thermal neutrons. The difference is we have a polyethylene plug that we put in place between our sample and the source when we're looking at this thermal neutron exposure. You can see here there are these back-to-back tritium and alpha particles that I've talked about on the thermal sample. And you can see that there are a lot more point sources on the fast neutron exposure. It's also worth noting that for both of these samples, we exposed them at our lab in Virginia Tech and then flew them to Zurich to measure them. So they had to undergo this flight where they were exposed to a lot of cosmic rays. So that's another reason why we wanted to build one of these microscopes at Virginia Tech.
What are the radii of the tracks?
The radii of the tracks?
Yeah,
I
roughly.
think I have a zoomed in one. Yeah, here. So this is one of these back-to-back alpha and tritons, and you can see, so this is 10 micrometers. The overall track is somewhere around 35 or 40 micrometers. So the width is probably, I mean, you can see there, it's maybe 8 micrometers or something like that. Yeah, so this is a zoomed in image of one of these back-to-back alpha and tritons from thermal neutron capture on lithium-6. The overall length agrees very well with predictions from SRIM. So you can see here, there's the triton and the alpha particle at those energies, and they produce these tracks that add up to somewhere around 40 micrometers or so. You also see this kind of increase in the density of damage at the ends. That's a very like Bragg-like peak that we see. We see that both in the TRIM simulations as well as in our data. The other thing that's worth pointing out here is that from the TRIM simulations, we expect to have a pretty low density of vacancies along the track itself. And the fact that we see some, lines in between these two peaks means that we do really have sensitivity to only a few color centers. So as I mentioned, so those results were all very promising. Over the summer, though, we built our own mesoSPIM at Virginia Tech. We swapped out the camera for this low noise single photon counting camera from Hamamatsu. The biologists use 2 different excitation arms because their samples are typically less transparent than ours, so they need to probe them from both sides. With us, our lithium fluoride or our diamond samples are all mostly empty space, so we can basically see them through them from just looking at excitation light from one side. We also then had some modifications to the software. There wasn't a way to track the temperature of the camera and then enable water cooling. The biologists don't really look at long exposures for their images, so we had to do some modifications for that and then add some additional metadata. And really, we just got this camera and the setup operating in October, and we're still kind of working on some of the characterization and the calibration measurements. So we're still very early on with this, but we do have the first images. And this is one of the raw images with our new camera. And you can see immediately, this is much nicer than the previous ones that I was showing. This camera has a lot lower noise than the previous one, and this is without even enabling water cooling. So we're expecting that the noise will decrease further, and we're excited to go ahead and take some large data sets with this setup and start looking at the analysis. There are some kind of weird shapes here and here. Those are just some reflections from the quartz cuvette, and we can deal with those a little bit by just rotating the sample slightly. It's not something that really is going to be a showstopper. Okay, so that kind of catches you up to where things are with our microscope at Virginia Tech. I wanted to talk a little bit about some of the ongoing things that we're working on. So one of the things is we're working on developing a simulation to make images that look like the mesoSPIM data, so we can then run our analysis scripts on them. We can do that to check and see if we're introducing bias in our analysis. We can use that to study the energy resolution of this, and also as input for machine learning training efforts. So what we're doing right now is we have a simulation that's developed in GANT4. It saves whenever one of these ions is created, and then we replace that with a track from TRIM that has these vacancies in it. And that's what we use to actually generate something that, if you squint your eyes, looks a lot like the data I was just showing. We then apply a point spread function that's measured, as well as camera noise to this. So we have this simulation up and running, and we're working on our analysis of the raw data and plan to run that on the simulated data as well. We're also looking into the sensitivity if we use this to look for reactor neutrinos. So we've been trying to model the backgrounds. If we were to take one of these samples and take it to the Leibstadt reactor, which is where this CONUS Plus measurement was, making the first measurement of sevens earlier this year. Shown here, this is kind of what the reactor looks like. The samples themselves would be about 20 meters from the core in the same location as the CONUS Plus detector. And with that, we looked at the CONUS Plus simulation effort and did our own simulation using CRY, which is a cosmic ray generator, and GANT4. And overall, we're able to produce a very similar result in terms of the backgrounds of different energy neutrons that we expect to be incident on our detector. They're passing through this different levels of shielding. We can do that to then estimate what our sensitivity is. So shown here, this is the recoils from our simulation that form a background as well as a signal. And you can see that we would expect with a pretty small exposure, around 30 gram years. So this is 30 grams of lithium fluoride at a reactor for 1 year. We'd expect to start to see some of this signal from this low energy sub-IBD neutrino scattering.
How many cubic centimeters of lithium fluoride is 30 grams?
Yeah, so the density is 2.6 grams per cubic centimeter. So it's only a handful of these crystals. And then, as I said, right, one of the advantages is that we don't need all of this active electronics. We can have a much simpler shielding design. One of the things we've talked about is just vacuum sealing these crystals and throwing them in a big tank of water. So you can do things like that. With this, though, we probably would want to bring our microscope nearby to do the imaging, on site, as opposed to then taking these up into a plane and taking them, for a significant amount of time out of the water shielding. So the mesoSPIM is something that you can, pack up and take with you somewhere else for these kinds of measurements. And that's one thing that we're talking about doing, looking at dark matter backgrounds as well. So here we've taken parameterization from May and Heim, where they looked at the muon-induced neutrons at Snow Lab. Their original simulation used Fluka. We're using GANT4, and we're able to more or less reproduce the same results. So this gives us a good estimate of what our backgrounds would be if we were to take these crystals underground to a site like Snow Lab. And we can look at what are the nuclear recoils that we expect to see in this during an exposure. Recently, we've begun using the MUTE generator to simulate cosmic rays. MUTE does a much better job at above ground sites like Grand Sasso, where you have a non-isotropic muon distribution. So that's one of the things that we've been switching to. With this, we're talking about trying to do a kind of active dark matter search rather than using these old samples that we've talked about. So we would take lithium fluoride to a site like Gronsasa or Snow Lab. We'd anneal it underground, do a scan with our microscope, expose it, and then do a scan a year later. And we think with this, we can set some limits or probe some parameter space for kind of lighter spin-dependent dark matter, and a very similar part of the parameter space as the CREST experiment that's exposing. So they also use lithium fluoride for their searches, and I think they're looking at something like 50 grams for that. Then finally, I've talked a lot about the fundamental physics applications for neutrinos and dark matter. There also are potential safeguard applications for this. Over the summer, we visited the National Criticality Experiments Research Center, or NCERC. We exposed several of our small lithium fluoride samples to neutrons from the Burke ball, which is beryllium reflected plutonium. It has about 4.5 kilograms of weapons grade plutonium. And with this, our goal is to measure the neutron spectrum emitted from this Burke ball, as well as study the directionality of the fast recoils using kind of these passive detectors as a proof of concept. You can see here, this is one of the lithium fluoride crystals. For this measurement, we went for some 0.5 centimeter side length crystals. And that was it was cheaper to get a bunch of those than the larger ones. Overall, though, the lithium fluoride is not especially expensive. I think it's somewhere around $100 or $200 per one of these centimeter sized cubes. Then we had some 3D printed holders, and we had to make some markings on our crystals so we could tell which direction they're actually oriented. So when we take these back to our lab, we know, which way they were pointing relative to the neutron source. And then finally, we started looking at some other materials other than lithium fluoride. So we have a couple natural samples that we've looked at the fluorescence from with our spectrophotometer. So shown here, this is a sample of Durango apatite, which has a very nice fluorescence emission around 525 nanometers. The downside is that we need 315 nanometer excitation light, and we're not sure if that will work with our existing optics. We've also looked at another material that's similar called phenakite and saw a similar kind of fluorescence behavior. We're also looking into diamond. So diamond is generally pretty well studied. And we're looking at radiation-induced fluorescence in synthetic diamond samples. So we got a small 0.5 centimeter by 0.5 centimeter by 1 millimeter diamond sample. We exposed that to a gamma source. We were able to get these two fluorescence bands that others have seen in the literature, around 575 and 660 nanometers. And then we actually had an opportunity. Someone was coming to Virginia Tech to show off their microscope, and we were able to do some images of our diamond sample after radiation in that. And you can see that there are these bright bands. Those are actually the grain boundaries of the diamond. So this sample had been annealed, and that causes these vacancies to move around, and basically they find the grain boundaries and get stuck there. So we're doing some more work into diamond, both with synthetic samples right now, and we hope to eventually look at some natural samples. And that brings me to the end of my talk. So to summarize, so crystal damage is a signature of nuclear recoils. We're trying to use this to search for interactions from neutrinos, neutrons, and WIMP dark matter. There have been a lot of past efforts to do that, but they're really limited by the amount of material they can scan. And we're hoping to get around this by using the SPIM to look for color centers produced from nuclear recoils. We use spectroscopy to first study the bulk fluorescence properties of these materials and figure out how it responds to different kinds of radiation. And then we've recently obtained the first images of damage tracks in lithium fluoride using the mesoSPIM at the University of Zurich. And we've completed our own mesoSPIM at Virginia Tech and have the very first commissioning data now. We're working on doing some calibrations of that, measuring the point spread function, making things production ready so we can start looking at real data in the next couple of months. And then there's a lot of other materials that we're interested in exploring. So with that, thanks for your attention, and I'll take any questions.
It
seems like there's a bunch of parameters that would dictate what sort of materials are like possible to use for like the mega year,
Mhm.
1 gram, like you said, the annealing and background radiation and the fluorescence profiles that are available, crystal profiles that are available. Is there a sense of like, I understand you're exploring a lot of materials because you're also making these to use as passive detectors and you can grow them yourself. But for the like naturally occurring ones, is there like a subset of like 1 or 2 types of crystals that seem to be like the best?
Yeah, so I mean, I think we won't know really if things are actually feasible until we try to image them. But part of this project involves consortia between an interdisciplinary consortia. So we have some geologists that we work with who are helping us to identify kind of what are the areas where you can find samples that meet the criteria that we're trying to go after. So yeah, it's something that I don't know a lot about, but luckily we have these geologists who are working with us for this. Diamond is, I think, one of the ones that's actually, even though it's expensive, it's relatively common. So that's one material that people are interested in because of that. So there's a couple groups trying to look at damage in diamonds right now.
Nice talk. Couple of questions. On the slide where you showed the sensitivity projections, can you remind me why this is a band there? I must have missed that.
Why there's a band here? I think this probably has to do with some uncertainty on what our actual threshold would be for these detectors. I'm not 100% positive that's what it is, but with the TRIM simulations, there's some scaling factor between the number of vacancies that TRIM predicts and the actual number of vacancies that you see in data. And it's a factor of like 2 or 3. I think that might be one of the things that goes into this, but I'm not entirely sure on what's setting the width of that band.
The images that you show in terms of track identification and so on, that's very striking, but what is, do you have an estimate for misidentification and the intrinsic background of the method or systematics in the method?
So there's a couple different approaches to try to tell what you're looking at. So we're exploring machine learning approaches, and we have both real data as well as simulated data sets that we're playing around with some different algorithms to do that. There's the idea that we have typically when people have looked at these kinds of track analyses, the thing that they focus on most is the track length. But you potentially get some PSD capability from looking at the track length as well as the number of vacancies. And then the other thing that this is a little bit speculative that I'm really interested in trying is that I mentioned we have these 2 different fluorescence bands. So I can get that picture back up. And we see different ratios of the fluorescence in the 2 bands. So that's potentially some other information that you can get out if you image the same region with a 650 nanometer bandpass and a 525 nanometer bandpass. You might be able to extract some other information about what caused the damage that way. Does that answer?
Yeah.
Okay.
Have you looked at sodium iodide or cesium iodide or something?
No, we haven't yet. Cesium iodide would probably be easier to start with just because it's of the hygroscopicity, but no, we haven't looked at that yet.
So you're irradiating the lithium fluoride with like cobalt 60, which obviously is going to leave like a pretty decent sized track, which you're able to resolve with what you've been able to work with. But how would you have to improve, or like that resolution probably wouldn't be large enough for when you're going down to the neutrino tracks and the dark matter. So how would you have to adjust this technique to kind of account for that, I guess? Like,
yeah,
so that was something we were struggling with too a bunch.
Yeah. So I mentioned this earlier. So it's pretty difficult to have a gamma actually create one of these defects. Most of the time, if you have a gamma in your material, it's going to interact with electrons, not the nucleus itself. And it's really only once you get to kind of higher energy gammas that you have to worry about this. We've looked at some samples that have been irradiated with gammas. I don't have any nice pictures of it here, but I do have an image that somebody has looked at with a wide field microscope, where they have the same sample with these nice nuclear recoils, and then they give it larger doses of gamma radiation. And basically, the gammas don't create tracks. What they create is kind of this background like haze or glow behind the samples. So here, this is the same image after exposing it to different amounts of gamma radiation, and you can see kind of this background forms, but you still have these tracks. One thing that's really interesting is that in some of our studies, we've seen that the fluorescence from gamma radiation actually anneals at a lower temperature than the neutron ones. So then we have this idea of trying to maybe bake these samples after exposing it to get rid of any gamma fluorescence. But generally, we think we have sensitivity to groups of 1 or 2 color centers. So that's all we need to see for something like a neutrino nuclear recoil or something like that. Is that?
Yeah.
Okay.
Yeah.
Can I ask so what limits you from just fluorescing these things? Do they bleach or something? Or could you just, like, if you find them, can you go back with a confocal and see every single color set?
Yeah, so bleaching is a material dependent property, and you can bleach these. Lithium fluoride is relatively stable to bleaching, diamond even more so. So yeah, you can image the same region multiple times. And then you could try, then we've talked about actually taking this to then a TEM or something like this to get a high resolution image of the tracks. There's a little bit of issues trying to make sure you're looking in the exact same region, but I think there are some ways that you can try to get around that. But yeah.
So I was a little confused how you handle the cosmogenic backgrounds. You mentioned putting it in a water tank, but without a timestamp, you can't veto. So it's just like
No, it's just shielding. It's not vetoing. Yeah. Huh? And like one of the nice things is that if we were to take this and deploy it to a reactor, we actually have a measurement of the thermal neutron flux from these captures, so we can kind of get a handle on backgrounds that way.
any other questions?
Yeah, if you're dealing with natural crystals, I imagine you're gonna have some level of impurity in any of these. I guess how do you control for that, or what level of impurity is acceptable when you're looking at the long term?
So in terms of like radioactive impurities, or in terms of
Or just impurity in the crystal structure?
Yeah. So that's something that I think we need to actually look at some of these natural samples to understand. We did try to image one of the apatite samples at the University of Zurich mesoSPIM. We didn't have quite the right wavelength of light to produce the fluorescence, but we weren't seeing, a bunch of defects and stuff in the images. They were just empty. So things might be, different once we actually get to the right wavelength to produce fluorescence if some of these defects are producing a similar type of vacancies, but that's something that we don't really know yet.
This is pretty off the cuff, but like I totally appreciate the interest in having like a passive detector. Is there any utility in kind of making this a dual readout as like a bolometer or a scintillator at the same time?
So one of the things that we've talked about doing is this crab approach for calibration. So with this, we're looking for lithium-7 in these crystals to capture a neutron. And when it does this, it emits this 2 MeV gamma, something like 90% of the time, and then 2 1 MeV gammas, the other 10%. And that gives this a few hundred EV of a recoil from the emission of this gamma. So we're talking about trying to do that. With that, we need to image this recoil before the lithium-8 that's formed decays, which is something like a 837 millisecond half-life. So we've talked about doing some kind of measurements where we have a different detector nearby to try to measure this gamma as it escapes, but we haven't looked at like instrumenting it with a PMT at the same time. Lithium fluoride is a scintillator, but I think it typically needs to have thallium doping to make it something that's sensitive to PMTs. So we've thought a little bit, but we haven't actually tried anything like that. I think there may be some interest in trying to do something like that and maybe collaboration with Tommy or something like that.
Any other questions? If you think of something later, maybe catch him in between his meetings, but let's thank Sam again.